look at the pink slime that's on your shower curtain, or you look at a mass of bacteria on a tube that clots it, whether it's a catheter or an industrial tube, those things, those communities are called bacterial biofilms. And several of my colleagues in science showed years ago that inside of those communities, bacteria are talking to each other. They're using small molecules to say who they are and to drive changes in the expression of genes necessary to control behavior. So the term social microbiology or sociomicrobiology was first created by Dr. Pete Greenberg and his colleagues in the 90s. And it's to talk about how do individual bacteria work together to make a colony or make a community. And now, for example, the gut is an area of community behavior that we study. So, for example, the gut microbiome, or the bacteria that live in your teeth, or the bacterial groups that live on the soil. All of those situations require bacteria to interact with each other, to coordinate, and now we understand also communicate. So my group in general is really interested in how do siblings talk to each other, and specifically, how do they share markers of who they are? How are they different than either their cousins or something really foreign? And then how does the communication of that identity work in terms of the mechanisms of cell-to-cell signaling, the changing of individual cell behavior, and then how does those interactions on the single cell level lead to group interactions? How do bacteria move over centimeter-wide distances as a whole community when a single individual can only go tens of microns, so 100-fold smaller? And we studied the role of identity signaling and communication in that.